The objective for today's experiment is to qualitatively test amino acid and proteins in order to describe their structure and properties. For the purpose, we have taken several amino acids like glycine, glutamic acid, lysine, tyrosine, gelatin, which is a protein, casein, which is also a protein, albumin, which is also a protein, proline and cysteine. In addition to this, we will require certain other things like the test tube rack, wash bottle and pH paper. In a labeled test tube, we will take 0.5 gram, approximately 0.5 gram of the amino acid. For example, this test tube is labeled tyrosine, so I am going to take around 0.5 gram of the tyrosine. As you can see, we have taken 0.5 gram of various amino acids and protein in the given test tubes. To the 0.5 gram of the amino acid, like in this case we have taken tyrosine, we are going to add around 2 ml of the deionized water. And similarly, we are going to add 2 ml of deionized water to all the test tubes containing various amino acids and protein. Mixture, we are going to put a stopper and it is to be shaken vigorously for 2 minutes. After shaking vigorously, what we observed was that glycine is completely soluble. There is no residue at the bottom. Lysine is also completely soluble, but lysine was available in the form of hydrochloride, so probably its solubility is a bit more than glycine. As we move further, we can see glutamic acid. It is only sparingly soluble in water. We can see the residue and it is not completely soluble in water. Tyrosine is it has even lesser solubility in water. As you can see, there is still a lot of residue remaining for tyrosine. As for the proteins like gelatin and casein, they are not found to be soluble in water. In order to test the solubility as a function of pH, we have taken the glycine solution from the previous part and now we are going to add 1 ml of 3M NaOH solution to it. After adding NaOH solution to the glycine, we are going to put the stopper and shake it vigorously for around 1 minute. You can see a slight turbidity has developed and now we are going to place the red litmus paper in the solution. Because it contains NaOH, it has turned blue. To this now, 3m HCl will be added dropwise till we obtain a red color for the litmus paper. You can see the color has started changing to red. You can observe that the solution has become clear again. As we had seen that glutamic acid was only sparingly soluble in water, we will now test the solubility as a function of pH. For the purpose, to this test tube, we will add 1 ml of 3m NaOH solution. We will stopper the test tube, put the stopper and shake it vigorously for 1 minute. As you can see, now the glutamic acid has become soluble. You can see a clear solution. To this we are going to add a piece of litmus paper. You can see the litmus paper has turned blue. To this we will add dropwise 3m HCl. We are going to add the HCl solution till the litmus paper turns red. That means the solution becomes just acidic. As the turbidity of the solution has increased a little bit, the litmus paper has turned red and the turbidity of the solution has increased a little bit for glutamic acid. So that means in presence of a basic medium, the solubility has increased, but in presence of an acidic medium, the solubility decreased again. The effect of pH on solubility for tyrosine. Tyrosine, as we saw, was almost insoluble in water. To this now I am going to add 1 ml of NaOH, 3m NaOH, and using a stopper we are going to shake it vigorously for 1 minute. As you can see, just after shaking a bit, the tyrosine is completely soluble. You can see it's a clear solution now. It is completely soluble when I added 3m NaOH to it. To this test tube now I will add the litmus paper. Because it is a basic solution currently, so the litmus paper has turned blue. Now to this we will add 3m HCl dropwise to see the effect of acidic medium on the solubility of tyrosine. You can very clearly see that the residue has started appearing again, indicating as it turned red, the litmus paper turned partly red only, and you can see the appearance of the residue indicating that tyrosine is insoluble in the acidic medium, whereas it was completely soluble in the basic medium. To the gelatin suspension, we saw that it was insoluble in water. To this again we will add 1 ml of NaOH and we are going to put the stopper and shake it vigorously to see if it becomes soluble as a function of pH. You can see that it has become slightly more soluble, or gradually it is becoming soluble. Even after addition of NaOH, the protein still seems to be not soluble. We have added the pH, the litmus paper, and let us now add HCl drop wise to see if there is any effect. You can see the litmus paper has turned red and the solubility of protein has also increased. You can see the protein is now almost completely soluble in water. As we know that protein undergoes acidic hydrolysis, so probably this is the reason why it has become completely soluble in acidic medium. For casein, again we will add 1 ml of NaOH to see if it is we stoppered the test tube and tried to shake it vigorously. As you can see, the casein protein is still turbid even after addition of NaOH. Let us now add litmus paper to it and then we will add HCl drop wise to see if there is any effect. So you can see we have added litmus paper and litmus paper has turned blue. Now we are going to add 3 m HCl drop wise to see if there is any increase or change in the solubility. You can see there is formation of some kind of turbidity. The litmus paper has still not turned red, so maybe I need to add some more drops of HCl. Yes, as you can see, the litmus paper has turned red, but the turbidity has increased with the addition of HCl. So it seems that casein was more soluble in the basic medium compared to acidic medium. As we can see the formation of turbidity, the solubility has decreased in the acidic medium, whereas the solubility increased in the basic medium. The solubility as a function of pH has different effect on different amino acids and proteins. For glycine, which was already soluble, it has not caused much of an effect. Lysine also remained soluble in both the mediums. For glutamic acid, we saw that it became more soluble in the HCl solution on addition of HCl solution. However, the opposite was seen in case of tyrosine. The solubility decreased as a function of acidic pH. You can see the turbidity on addition of HCl. For gelatin, the protein, we saw increased solubility in acidic medium. Whereas for casein, we saw decreased solubility on addition of HCl. So, we can say that pH affects the solubility of amino acids and proteins.